So my name is Sunil Agarwal. I'm a physician, a resident at New York University Medical Center. I'm doing my residency in physical medicine and rehabilitation. I did my PhD briefly. A little bit of a timeline. Steph asked me to give a little bit of a picture of this. So let's dig right in. Just these two disclosures we generally give at medical conferences, given the influence of industry and pharma. So I want to state clearly that I don't have any financial ties with any commercial interest, and I will discuss FDA non-approved uses of drugs, which happen all over the place. That's very commonly said in talks in medicine. This is the data. So then essentially that's 2.3 publications a day for the last 20 years if you wanted to do that out. That's 24,000, and then in 2013 it's projected to be 2,689 based on where we're at now per year. This is what's going on. Since I'm the first sort of person putting up slides, I had to show maybe some folks, maybe Dr. Schwartz says he's never used marijuana, he was at Berkeley in the 60s, so in case you didn't know what it looked like, this is a little bit of the pictures of its life cycle from seed all the way to production. This is fields in Morocco, and people publicly using cannabis in state houses and so on. Other big developments in cannabinoid science have been in the understanding of the molecular structure. This is not down to the subatomic string theory level, but at least it's down to the molecular level where we can see. For a long time you just saw the crystals and resin, but now we have electron microscopy pictures showing the showing cannabinoid, where cannabinoids are produced in the plant, and we have a greater understanding of the different stages going from the leaf, how the resin is secreted, and what portions of these matrices where cannabinoids are synthesized and released. And I won't go into the details here, but you can see it's been really well worked out. And then, moreover, the genome of cannabis was sequenced in 2011. Every base pair for two strains of cannabis, a traditional hemp strain and a traditional medicinal strain, have been sequenced. And this is just a huge increase in our understanding of down to the molecular genetic level, what cannabis is and how it's working. So then the other history piece is the fact that in 1840 was the first sort of modern scientific approach by a doctor to study cannabis by O'Shaughnessy in India. And then after he showed that it was useful in tetanus, in spasms, and rabies, a few other conditions, things that, basically based on hyperexcitation of the nervous system, he was able to publish that, and it was he was eventually knighted for his laying the telegraph throughout India, but this other work of his was very important and led to, I've seen more than 200 different medicinal cannabis preparations that were in circulation in the United States and Europe. Menstrual cramps, asthma, cough, insomnia, labor pains. And even the father of internal medicine, William Osler, who was at Johns Hopkins and invented the idea of rounding, he's the one that said that for migraines there was no superior treatment. So this was collected empiric knowledge from a whole 100 years of medical use and pharmaceutical distribution of cannabis. Then there's also these other histories with the Mexican laborers in port city of New Orleans, where cannabis was brought into the sort of popular culture through the jazz scene. I'm sure most of folks are familiar with this history. So, and then as Dr. Woodward was mentioned by Dr. Schwartz in 1937, did testify at this Marijuana Tax Act. And what's interesting to note is that this was around the same time Nobel Prizes were being given out in the discovery of neurotransmitters and in the discovery of actually electroconvulsive therapy. Those were two Nobel Prizes in medicine that were 1936 and 1938. And in 1937 we have doctors testifying, not Woodward, but his counterpart, that marijuana caused dogs to go insane based on his research at Temple University. So the AMA's position essentially was that future investigations may show substantial uses, but one thing that folks don't realize is that the AMA also said that there were already accepted uses that they had that there was no replacement for, and I wanted to just mention that. And Medicine later followed up on this. So this is from the testimony of Woodward. He said that there are exceptions to which cannabis cannot be apparently successfully substituted for. And Woodward said that, are there any uses for, so essentially he says that cannabis can help reveal the psychological, psychotherapeutic, it could be useful for psychological and psychotherapeutic research to revive hidden or old memories. And that's what psychoanalysis depends upon. And he said that cannabis, there was no other drug that medicine had that could do this. So it's a form of sort of research and development that doctors basically put on testimony to Congress that cannabis was useful for this purpose. It was written about in JAMA in 1935. Said here about the authors conclude the drug has some value in revealing the content of the subconscious mind. This was the understanding in medicine in the 1930s. So the upshot of the Marijuana Tax Act, what came out of it was this definition of marijuana. And I just wanted to highlight that this is still the definition used today in federal law for marijuana. And just note clearly here that any resin extracted from the plant, every compound manufactured salt derivative is considered to be marijuana. They were very careful in crafting this definition. Between 1937 and 1970, when the Controlled Substances Act passed, there were actually researchers like this gentleman. This is kind of a goofy artwork here from a marijuana review piece, but this was the psychiatrist, Dr. Herman. He actually had he was a duly appointed researcher under Class 5 of the Marijuana Tax Act, which means that he was able to prescribe marijuana for his use in his psychiatric practice. And in this article, he described his interest in using cannabis as a psychoanalytically oriented psychotherapy adjuvant. And these are just some of the things that he found in his research, that it could be used as an emollient, lowering conscious censorship, and et cetera, et cetera. So this was kind of another form of research that was going on before the 1970 law was passed. The modern marijuana pharmacopoeia includes these cannabinoids and cannabis-based medicines. Well, I won't go too much into detail because of time issues, but you can just sort of see here, this is the cannabis extract that I was talking about. These are cannabis cigarettes from the federal farm. Why I brought up the issue about marijuana's definition is because you actually have this is the patent application for the production of the cannabis extract, the second one I showed on the slide, which goes by the name Sativex. And it's important to note that this is produced, a botanical drug substance produced from medicinal cannabis. And this is just important. The reason I bring this up is this sort of helps you to understand that whatever work is done on this medicinal cannabis extract does apply to our understanding of marijuana's scientific medical evidence base. So this was also these are quotes from their patent application from 2008. And then also Dr. El-Zali, who has the federal farm that grows the cigarettes, the cannabis cigarettes that I showed you on that picture slide, he told on his record before the DEA that he was permitted to grow marijuana to extract THC for sale to this pharmaceutical company to manufacture generic Marinol. So all of these things, remember that what's in a name, any compound, any resin, any of salt, derivative, mixture, or preparation of the plant marijuana is marijuana. So keep that in mind when understanding the, and I thought I'd put Shakespeare's Romeo and Juliet, 1600, what's in a name, that which we call a rose by any other name would smell as sweet. So I don't know how much time I have left, but quick mention that most of the research that was done before the 1990s was non-controlled clinical studies, but they had large numbers of patients, 748 patients who smoked cannabis in these 7 state-sponsored clinical trials for cancer patients getting chemotherapy. These were the states that were involved. And 70 to 100% relief from nausea and vomiting was self-reported by patients in these uncontrolled but cannabis versus THC pill studies. And then essentially though, after 1998, when the first FDA-approved clinical study of cannabis in a patient population was enrolled its first subject in 15 years, is sort of what catapulted a more modern cannabinoid research trials. And essentially, if you go back to 75 to 2009, you can add up 110 trials involving 6,100 patients, and that includes cannabis and cannabinoids in human studies. In the United States, 33 to 40 different inhaled clinical cannabis trials, about a third of which were gold standard, that is double-blind, randomized, placebo-controlled, were done in this period. So those 33 trials looked at appetite stimulation, HIV neuropathy, other forms of neuropathic pain, spasticity, weight loss, glaucoma, asthma, vomiting, and nearly all showed benefits favoring inhaled cannabis. You can even do things like this, where you can stand up all the different human trials for nausea and vomiting. And these are called meta-analyses, systematic reviews, and you can show that you can add up all trials and show that they favor treatment. This is kind of a higher level of evidence-based medicine that kind of proves this point. This is 18 studies of cannabis and cannabinoids that were reviewed. So the rest of this talk, I just sort of showing, it was how do you do a history of medical cannabis research without sort of saying, hey, these review articles showed, these hundreds trials showed this benefit. This review article had 18 randomized controlled trials for pain with 766 participants in total. 4 of the trials were inhaled cannabis. The other ones involved cannabinoids and cannabis extractions. But all of the high-quality trials and the inhaled cannabis trials found a positive effect with no serious adverse effects, these authors wrote. They're from Canada. So, and they also said that, so their conclusion was cannabinoids are modestly effective and safe option for chronic non-cancer pain. And given the prevalence of pain, and we need additional treatment options in this field. Another study said that when you looked at the use of treatment for this condition, HIV-associated sensory neuropathy, it turns out that cannabis is essentially one of the only drugs that has an solid evidence base to treat this condition. And so it's not just that cannabis is shown to be effective, you can pick a disease and you can find out that only cannabis has the most solid evidence base to treat that disease. So it should essentially be the #1 recommendation to treat. And so many how we look at trials, the number of patients, how they're designed, efficacy, adverse events. And I can't show you all, there's so many, I just can't show you all the details. But this is a picture of how the trials are done at San Francisco General Hospital Inpatient Clinical Trials Ward. You have a pharmacist dispensing the cannabis, you have a patient in a controlled setting consuming it, and with a sort of puffing paradigm, and you have a pain scale, the visual analog scale, where to tell you what their pain score is before and after. Neither the doctors doing the study nor the patient knows if they have active or placebo cannabis. And at the end, we all find out, and you can do a graph like this and show that pain scores go down in the active trial, and they don't go down in the placebo trial as much. And that's kind of evidence, scientific evidence, how you can interpret these kind of studies. Another study, again, I'm just showing you the evidence, was to treat spasticity in a double-blind controlled fashion, and essentially this is showing. So the patients who use cannabis first, this is where they started up here, and this is where they drop to after their treatment, their spasticity scores. This is where they drop to after their treatment, like this. And then, this is before and after testing of spasticity in multiple sclerosis patients. And then so you can kind of summarize the data in this way over here too, that before and after, when you have cannabis, the patient's level of spasticity drops down to here, but if they have placebo, it stays up here. This is kind of where the rubber meets the road. This is where the money is. And anyway, I'll skip all this. And just to sort of know that the cannabis extract, various cannabis extracts. This is one done with Canador, another cannabis extract that in large trials with 666 patients. 660 MS patients at 33 sites, you can do a 12-month study and show that you can have improvements in mobility, which means that patients have less disability in MS and less emergency room visits, less hospitalizations. This is the kind of evidence that exists for when we consider this also equals marijuana, which it does according to the definition. Also, this is the one involving nabiximols, Sativex, And this showed also in a first phase 3 placebo-controlled study of the efficacy of the endocannabinoid system modulator, which is essentially this, the US applied name nabiximols, but in any other country or if in a different context that would be considered marijuana. So you can see here that they show an improvement at 10 weeks with from baseline pain scores in patients who have neuropathic pain from multiple sclerosis. And apologies to folks who know this data much better in the audience, but I'm just trying to give an overview of what's out there in the history of cannabis research. So there's even patents that the US government has. I'm not going to go into that. And then the last thing I'm going to say is that because of the blockades and the difficulty of doing research with controlled studies, a lot more research is coming out in the social science literature looking at cannabis as a system, medical cannabis programs, and the patients who are using those programs, community-based research. And that's some of the research that I've done as well, and I can talk about later, and some of the research you'll hear later today from the speakers that come after Dr. Ware and I. So thanks for your time, I appreciate it.
Good morning, everyone. It's a great honor to have been invited here. Thank you, Steph and the organizers. Nice to come down from Canada, where it is even colder than it is here. Don't complain, it's -20, or it was yesterday in Montreal, so that's why we're so good at ice hockey. Or were. Okay, so my job is, following two very, very informative sessions, is to try and sort of dive into the endocannabinoid system a little bit and perhaps provide you with some of the tools that we need to understand exactly how this drug appears to work the way it does. Most of what Sunil showed you in terms of the increase in the studies that have been conducted and the research that's been published in the last 50 years on cannabinoids has been on the endogenous cannabinoid system and exploring how it works and why it's so important to human physiology. It's important to point out also that in spite of the NIDA restrictions on cannabis research and access to different plants, the research that you've seen from Sunil, the clinical trial, especially in the last 10 years, have continued anyway. And that's a testament to the kinds of people who want to do this research. They're willing to keep trying and keep pushing and keep doing it in spite of the barriers. I did a study in Canada a few years ago where we had to get a placebo cannabis, and the only place we could get it was from NIDA. It took 2 years to import a placebo cannabis from the United States into Canada because it was handled as a controlled substance. It was still cannabis, it just had all the cannabinoids taken out. But 2 years of protocol reviews, NIDA, DHHS, DEA, FDA, everybody had to review it and sign off on it. It took 2 years. And then Christmas Eve, 2006, I received a package of 0 THC cannabis from NIDA. But to point out that it's possible to do the research. You do have to have a tremendous amount of stamina, and that in spite of the suppression, perhaps, and the unwillingness to contribute, the work has been continuing. And so it's worth a nod to the researchers who do this work, that in fact it can be done. So we have a desire. Is this thing going to work? Can you just move the next slide? So what I'd like to try and do is just tell you briefly in the next 10 minutes what the components of the endogenous cannabinoid system are. It's just very useful when you're speaking to clinicians particularly because they like to know what the mechanism of action of cannabis is. Very often you hear, well, it just makes people high and they don't care about their problems and that's why it works. It's actually a lot more than that, and hopefully by detailing some of the system, it'll give you some tools to use when speaking to clinicians and other health professionals. And then perhaps to summarize some of the pharmaceutical strategies that are being looked at to try and develop this system. So just perhaps to put it into a bit of context, again, Sunil has shown you the historical development of cannabinoids over the last 50, 100 years or so. It's probably worth positioning this, and what I've done here is to overlap it with the opioid development. And remember that opioids and morphine were used for thousands of years as painkillers, and it was really only in the 1800s that people identified the active ingredient ingredients. Then it was identified there were receptors, and now it's almost common knowledge. You go out on the street and talk about endorphins. Everybody knows what they are, body's own natural opioids, right? And we have, a family of pharmaceutical opioid drugs that are being used to treat pain and to treat various other symptoms. And in parallel to that, the cannabis is very similar, starting off as a herbal compound, identification of the active ingredients, identification of a natural target for it, and then subsequently, and where we are now, development of possible compounds that can harness that. We're just about 30 years later than the opioid story. Everything's shifted over to the right. But it's an important parallel, and it's a way that we can think of cannabis development as simply paralleling what happened with opioids. Next slide, please. The difference is opioids kill people. Okay, the USA consumes 80% of the world's opioids. It is an incredible volume of opioid medications that are consumed. The number of opioids that are being prescribed in the US have increased in the last 10 years, and this is data from the CDC, showing you how the number of prescription opioids, and I think this graph here shows you the opioid sales increasing dramatically over 10 years. This is the number of treatment admissions for opioids in the same period, and this is the number of treatment deaths. Opioid pain-relieving deaths per 100,000 also increasing. So it tells you two things. One, I mean North America, I'm not just going to pin this on the USA. As a Canadian, we have a drug problem, and it's opioids. But we also have a pain problem. We have a tremendous amount of pain in our society. And it's a huge burden. It costs us money in treatment, it costs us money in lost productivity, and it costs us personal suffering, as you heard from Professor Schwartz and his wife. And as a pain clinician, and as a primary care practitioner, I can tell you that the hardest thing you can ever do is to tell somebody, I've got nothing left to offer you, when they're in pain and they're not necessarily palliative, but they're going to live with this pain for years and years to come. That is a devastating thing to tell somebody, and it's even more devastating to have to hear. So, why are we interested in cannabinoids? Well, in Canada, may I have the next slide, please? I don't have data for the US, but I can tell you that in Canada, where we have a federal program and that we now have patients who can access and authorize cannabis for medical purposes legally under a federal program, we've seen a huge increase in the amount of cannabis that's, the number of people who are authorized to possess cannabis. Now, I don't mean to suggest that this is a huge increase in the number of people using cannabis. Most of these people were using it anyway. They've just now become part of the legal system. But what this offers us is an opportunity to now actually study these people. We know who they are. They're getting legal cannabis from a legal source. We have an opportunity to track and monitor that, and it's being missed. We're not doing that now. There's no system in place to monitor and track patients legally using cannabis over the long term so that we can be reassured that the drug is as safe as we think it is. That's a critical opportunity and something that we should be really focusing on. Next slide, please. So why do we think cannabis is much safer than opioids? Well, the main reason comes from this graphic here, where we know that the cannabinoid 1 receptor, the main target for THC, the active ingredient in cannabis, is incredibly ubiquitous in the human brain. It's one of them, in fact, it is the most common receptor in the human brain. And it's clustered in major areas where you expect to find a system involved in mood, in pain control, in muscle spasm, in appetite, in nausea, all of the things that we know cannabinoids are effective in. In dark green are the clusters, cerebellum for movement disorders, the amygdala here, very important in fear extinction, and the PTSD story, very tightly coupled with the prefrontal cortex. There's some really cool stuff going on there. But what you don't see is any cannabinoid receptor here in the midbrain, which is where the opioid receptors are found, and which is why in high doses opioids kill people. They simply stop you breathing. Cannabinoids don't do that, and we don't have this massive safety issue. So if we're looking for new pain control methods, if we have a pain problem, we don't want to be using opioids as much because they have problems. Why aren't we looking at cannabinoids? Well, so the story is that we are, and I think that's probably really what you heard from Sunil, is that there are huge attempts to try to explore and provide some data on cannabinoids. Next slide, please. They're not just found in the brain. Cannabinoid receptors are also localized in the periphery, so in the body, in the urinary bladder, which is probably why the drug was effective in treating Professor Schwartz's wife, in the GI tract, Receptors are found in the endothelium, they're found in the testes, they're found in the ovaries. The cannabinoid signaling system, which I'll tell you about in a minute, is probably the most ubiquitous physiological signaling system in the human body. No matter where people look, they find evidence for some kind of cannabinoid involvement in modulation of some form of activity. So it's playing a very, very important role in all of us all the time. We're all constantly synthesizing, metabolizing cannabinoids to function and to manage our everyday life. This is probably best illustrated by the fact that a few years ago a drug company produced a drug which blocked CB1 receptor, rimonabant. And this was done to, believe it or not, the clinical trials were very strong, and they showed that if you took one tablet of rimonabant every day for a year, you could lose 20 pounds. Okay, who's not going to want to lose 20 pounds? This was their blockbuster drug. It was approved in Europe, but within 2 or 3 years, they started to notice increases in depression and suicidal behaviors in patients taking it. So shutting down your CB1 receptor system is probably not a good thing. The drug was taken off the market and it was never approved in the US. And as far as I know, that line of research for pure CB1 antagonists has been stopped. There are still attempts to try and play with it, but that attempt has been stopped. So we need CB1. So just to show you how opioids, really, they bind to a specific receptor. The receptor has effects intracellularly which change the way that the cell membrane functions, and that results in its effect usually on pain modulation. Next slide. So cannabinoids work very similarly, but there are some really important differences. The main difference being that the cannabinoid receptor is located on the presynaptic end of a synapse. That is to say that a message coming down from here usually releases a transmitter which crosses the synapse, binds to the second neuron, and then that message is continued down the chain. And this happens all through the brain. But what happens when this receptor is activated is the neuron synthesizes a cannabinoid compound here, which rolls backwards to the CB1 receptor on the presynaptic terminal, which then suppresses further release. So it's a feedback loop. It stops the nerve from continuing to fire at will. And this is really important. And if you think about clinical conditions where nerves are firing at times when you don't want them, epilepsy, neuropathic pain, muscle spasticity, one could think of anxiety, even some forms of psychosis. There's abnormal, aberrant firing of neurons. A cannabinoid is the perfect drug to try and suppress that abnormal activity. And we think that in many of the clinical conditions where cannabinoids work, it's simply this balancing of neurological activity. And this could be an activator or an inhibitor. So you can suppress inhibition, which results in excitation. You can suppress excitation, which results in inhibition. It's a complex math, but essentially this is a modulating effect. And so the endocannabinoid system is constantly functioning to keep all of our nerves firing nice and smoothly. It's like the oil in the engine. Next slide, please. Another important thing to realize is the endogenous cannabinoids that we're using and making all the time are not proteins. Classically, we tend to think of neurotransmitters as protein compounds. These are actually lipids. They're fat-derived compounds. And the endogenous cannabinoids are derived from the interior of your cell membrane. So this is basically the plastic bag that wraps around every single cell in your body, including nerves, including the white blood cells, red blood cells. They all have a membrane. And in that membrane are these clusters of fats. And these are the fats that are derived from the foods you eat. So the omega-3, the omega-6, these are all metabolized and packaged and put into your cell membranes. Incredibly important source of fat. And the endogenous cannabinoids are taken from these little fatty chains and broken up and then released into the synapse. And that's how they have their effect. And then they're taken back. Most cannabinoids are extremely fatty substances. They absorb into fat. Brain is very fatty because of all the nerve cells. So this is why cannabinoids stick around in our bodies for such a long time. And in fact, the way that these two major endocannabinoids, anandamide and 2-arachidonyl glyceride, act on the CB1 receptor actually by getting inside the cell membrane and then binding into it. They don't sit on the top like a lot of proteins do. They have to get in here, and in that little pocket, they then activate G protein, which results in the changes in the nerve cell conductivity. So some really intriguing facts about the endogenous cannabinoid system which differentiate them from opioids. Next slide, please. I can't even read this from where I'm standing, so I don't expect you to. But what this is meant to demonstrate is that we've now characterized the way that these endogenous cannabinoids are not only manufactured, but also broken down. So there's a complex biochemical system by which those fatty acids are taken from the cell membrane, made into cannabinoids, anandamide here and 2-AG here, and then broken down again. Special enzymes that do this, there are special processes that seem to work. There are still some major chunks that we don't understand. But these are the major compounds. And this system is of extremely high interest to pharmaceutical companies, because they're looking for the new big thing. They're looking for new treatments for pain. They're looking to make big money. And sure enough, this system, because it's so ubiquitous, is a perfect target for them. So you can rest assured that every major pharmaceutical company in the planet is looking at cannabinoid receptor agonists, antagonists, endocannabinoid signaling compounds. They're all hunting. They may not say it publicly, but they've all got their labs, and I've spoken to many of them. Next slide, please. Let's not forget about CB2, the second receptor. We don't really know much about how this one works, but we know that it's tightly coupled to some of the immune functions. And so we can see here immune cells, which are, as well as neuron, these are the CB1 receptors. But here on the immune cells, CB2 receptors are increased in inflammation, in neuropathy. They're very involved in release of cytokines and the signals that tend to suppress, that lead to inflammation. And we know that CB2 is coupled to this. So very strong evidence coming through in the endocannabinoid system in the role it plays in inflammation. And that's not just the inflammation you get from a bee sting. There's also inflammation in the brain when you have injury. So neuropathy, you have a number of other conditions where there's actually very strong inflammatory changes inside the brain itself, and these little macrophages get in there and can also help clean up damage. And there seems to be some really intriguing work on cannabinoids and neuroinflammation. Next slide, please. So, very powerful system, like a beautiful horse. It's a wonderful thing to see and to know, but it's a difficult thing to control. So what are the techniques that are out there now to help us try to use this cannabinoid system in therapy to help patients who are suffering? The endogenous cannabinoids we've heard about, the phytocannabinoids, THC, cannabidiol, combinations of these are either in extraction forms, various other delivery systems. Synthetic cannabinoids are out there. Next slide, please. The endogenous manipulation of the inhibitors. These are the endocannabinoid molecules, so inhibitors of the enzymes that break down cannabinoids, methods to try and stimulate production or inhibit the uptake. That's all work that's continuing at the moment. Next slide, please. And work going on the target. Some of these compounds, the endogenous, don't just bind to cannabinoid, they bind to other receptors, and there's a tremendous interest in how compounds can bind not only to CB1 but also to TRPV1 and other channels, and these could potentially also be therapeutically active in other pain conditions, so even broadening the scope. So some novel approaches to how to get cannabinoids into the body is really intriguing, and we've probably heard, and some of you may be working on some of these, but oral preparations, edible products, tinctures, and so on, packaging THC into microspheres so they can be inhaled properly, topical delivery systems, creams, oils, even skin patches, buccal mucoadhesive preparations. You stick it onto the inside of your cheek and it releases THC very rapidly. That's work going on in Montreal right now. Vaporization, aerosols, portable ones, the big desktop type devices. There's even, I think Mahmoud El Soli has a patent on a rectal THC, so probably popular in Europe, not so popular here. And while the endogenous cannabinoid system seems to be a very powerful, a very important, and a very intriguing system for controlling pain and other symptoms, there are ways that we can use this that don't involve taking any drugs at all. Running raises your endogenous cannabinoid tone. There's been work by John McPartland suggesting that osteopathic manipulations raise your endogenous cannabinoid tone. I'm willing to put money on the table if anyone wants to bet me that yoga, massage therapy, any form of complementary therapy that makes you feel that mild euphoric feeling when you've had the treatment is an endocannabinoid effect. So there are ways that we can target endocannabinoids without using any drugs whatsoever. It's not just about taking a plant and smoking it. This is a tremendously intriguing, tremendously important system. Thank you for your attention. Enjoy the meeting.